Hi, everybody. Welcome to Chapter 8. In this chapter, we're going to be talking about genetic engineering. These are our learning objectives, so keep them in mind as we talk through the content. First, we'll start off with discussion about recombinant DNA technology. This is basically anything where we're using DNA for investigative purposes. So either identifying an individual, identifying genetic mutations associated with disease, fixing those mutations, changing them, or investigating them on a molecular level using other organisms as like a means, as like basically a platform for studying those genetic mutations. We don't always have to study that genetic mutation in the organism that it was derived in. It might be difficult to study genetic mutations and how they affect people in people, but if we can put those genetic mutations into play in yeast or something like that, now we can study the effects of mutations outside of that. So all of that falls under the realm of recombinant DNA technology, and we're going to talk about some of the systems that we use in genetic engineering to study these things today. So first things first is understanding kind of a little bit about DNA. We remember that the DNA double-stranded ladder basically is held together by hydrogen bonds. And so heat can actually destabilize those hydrogen bonds that are holding those two strands together and then separate them. And then whenever we cool the DNA down, it typically favors refolding back up and rejoining those hydrogen bonds back in the original formation. Especially whenever we have a lot of nucleotides there, so that we have proper, rebinding and things like that. So we can use this mechanism for investigating DNA, because once we can separate those strands, that gives us like two templates to work with. And a lot of the mechanisms that we use to study DNA basically kind of rely on separating those strands out and then repairing those strands somehow. We can even introduce mutations as a result of that. So some of the stuff we're going to talk about today, we'll get into those concepts. It's also important to understand how we measure the size of DNA. DNA is measured as base pairs. So here, this is base pairs, BP is how we would refer to that. So base pairs, basically like if your DNA strand sequence is A, C, G, G, C, T, then the other strand would match up with it. T, T, G, C, C, G, A. Those are the complementary bases, right? And remember that typically we'd have one of them 5' to 3', and the other one's oriented in the opposite direction based on those sugar phosphate backbones. This is just an abbreviated look at that. Okay, so anti-parallel strands here, and then this is one base pair. It's a pair. The A is paired with the T. So that's how we typically look at that. And if I was to say that the strand was AACGGCT, it would be implied that the other side of that would be there. So we typically look at DNA this way. We'll talk a little bit about some of our enzymes that are used to cut DNA here in just a moment. But an example of these endonucleases, restriction endonucleases that cut at sites that we call palindromic sequences, they cut at sites that are anywhere from 4 to 10 base pairs in size. To put that in perspective, the average gene for E. coli is about 1,300 base pairs. The human genome has 20,000 genes, which is a composite of about 6 billion base pairs on 46 chromosomes, 23 pairs of chromosomes. And I'm just giving some relativity for size of DNA here. If you see a symbol like this, KB is kilobases, kilo is 1,000. So already if we say 1.3 kilobases, that is 1,300 base pairs. So it's multiplied by 1,000 there. So add on the appropriate zeros as you would need to. So this puts that into perspective and that size. And we'll talk about this in the lab as well, but it doesn't come until almost lab 10, which will be past the unit 2 exam, about the same time as unit 2 exam, I guess, the lab midterm. So that stuff comes around the same time. So that's coming in time, but just keep this in mind as you're learning this content, that this is what we're talking about with the size of DNA with the base pair. So do be able to convert between base pair to kilobase pair to megabases. Megabases is million. So it's 1, 1,000, a million. And so each of those is 3 zeros added into that factor. All right, so first things first, we're going to talk a little bit about CRISPR. CRISPR stands for, there's a video, sorry, I didn't mean to click that far. There's video that I'm not going to play in this lecture because it doesn't play through appropriately in the recording. So if you want to view the video that summarizes how CRISPR works, the video is there and I do recommend that you watch it. So you'll need to watch it in the slides. So be sure you play that video in the slides. I do recommend that you watch it. It just doesn't carry the audio when I play it on the recorded lectures. Okay, so CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. And basically, these are short lengths of DNA that are recognized by bacteria and archaea as being foreign. And so they can kind of program themselves to see this sequence of DNA as foreign, and then that triggers them to cut it out. It's essentially a way for bacteria to protect their own genome naturally from invading, genetics. And so that would be from phages and foreign plasmids and things like that that might have DNA sequences that they don't want in their cells. So that's what CRISPR naturally exists for. And again, there's a video about it that covers it a little bit more clearly that I'm not going to play here. Moving on to restriction endonucleases. So CRISPR is like a more advanced version. With CRISPR, you have to know the sequence that you would want to cut at. The idea is that in genetic engineering, you could program CRISPR to cut at any sequence, but you have to pre-program it that way. And that would allow you to cut DNA at any sequence. And then potentially, then you could insert new genes and things like that as a response to that. That's not working really in advanced mammals right now. It's not really effective in advanced mammals right now, but the idea is that this could allow us to change anybody's genes anywhere, as opposed to restriction endonucleases, which has governed genetic engineering for a long, long time and is like the original big daddy of how we make changes in genetic code. So what is a restriction endonuclease? They are enzymes that clip DNA at specific sequences. So there are endonucleases that we might call EcoR1, for example. So this is the number 1. So EcoR1, it's from E. coli. It is restriction endonuclease number 1. And it might cut at, and I don't remember the exact sequence, but something like GGC, GCC. 6 nucleotides. And then, of course, the corresponding, again, this is always referring, if I say this sequence, this is one side of the strand, and the other side of the strand would look like CCG, CGG. This is a palindromic sequence. And so the next slide is going to talk about it a little bit more, but just to give us a little bit of reference, this means that the enzyme is going to typically cut here, all right, and it will create sticky ends, meaning that this end would be free and then this end would be free. So I'll kind of redraw it in the next slide, I guess, but to give a little bit better reference. And that if you have one free tab of single-stranded DNA, it wants to match up with another free tab. It could just match up with itself, or it could match up with a sequence that you designed that has the same sticky ends. So this allows for sticky ends to match up with each other. So that allows us to insert DNA. But EcoR1 only cuts at this sequence. If you want to cut at a different palindromic sequence, then you would need a different restriction enzyme. So they are very controlled on where and how they can cut. The enzymes already pre-exist. We cannot engineer them to cut somewhere else. They just already exist to cut at those locations. These are made by bacteria for a very similar reason, to cut at foreign DNA and plasmids and phage DNA and things like that to protect the bacterial genome. Similar function is what we see with CRISPR, but they work in different sequences. So here, these are set palindromic sequences that if that organism sees that, it can cut at that palindrome, create sticky ends or blunt ends, which would just be a straight cut. But we typically use the sticky ends in genetic engineering. So often these happen at palindromic sequences. So again, I'm going to move on to this, but I want you to keep in mind what we're going to be talking about with these palindromic sequences. Maybe the next slide will show it better, but it kind of does. So basically, these are sequences that are anywhere from 4 to 10 base pairs long, and they create overhangs that are single-stranded that want to match up with another single-stranded complement to it. Why do we call them palindromes? Because from the 5' to 3' direction on that first strand, they read the same way as they do in the opposite direction. So let me show you an example in this image here. So here we have and it does show us EcoR1, the correct sequence for EcoR1. And you don't have to memorize these. There are hundreds of different restriction endonucleases. You don't need to know what the sequences are that they recognize. You just need to know generally what these guys do. And so, for example, with EcoR1, you can see here that this is how this would cut. It would create sticky ends. What do I mean by sticky end? These overhangs here. So CTAG wants to meet up with GATC. And what makes these a palindrome? If we are talking about with EcoR1 here, you can see that it's GAATTC in the 5' to the 3'. So the other direction is GAATTC. Wouldn't you look at that? They read the same in the opposite direction. Additionally, if you take one strand and you fold it over onto itself in the middle, it matches up. So this is what makes it that very special palindromic sequence on how restriction endonucleases work to cut DNA. They recognize it. It goes the same pattern going forward as it does backwards. It reads the same because of the way that those nucleotides are set up and their complement on the other side. And so you cut there, it has an overlapping sequence on from one organism, that same overlapping sequence on a second organism, and now the sticky ends can match up if we mix those DNA segments together, and then we can seal them together with DNA ligase. Remember, DNA ligase seals together DNA fragments, creating those phosphodiester bonds and repairing them. And then this would allow us to insert genes into vectors. So this requires us to understand a little bit about vectors, but first we've got to understand these fragments that can be made, because even your DNA right now just randomly has some of these palindromes in it, just because that's just what the sequence is. And so my DNA, when I cut it with EcoR1, might make certain fragments just from those random palindrome sequences that EcoR1 recognizes. But yours might be different because you have a DNA sequence that's different than mine. This is referring to restriction fragment length polymorphisms. I know it's a mouthful. Okay, so restriction fragment length polymorphisms. The idea that if I cut my genome with EcoR1 and your genome with EcoR1, we would end up with a bunch of fragments of different sizes. And that gives us kind of a fingerprint of our DNA that can be used to essentially predict like who done it. My fragment sizes would be only for me. Your fragment sizes are only for you. And this is very predictable. We can actually use the different fragment sizes that are made by each person to help with identifying people that way, like in CSI and things like that. So this is the idea of different fragments that can be made by using those enzymes. All right, so restriction fragment length polymorphisms, cutting at that specific recognition site for those endonucleases and giving those different fragments that will differ from person to person and allow us for identifying them. So it's not just for cutting and then piecing together DNA, which is one useful tool, but also for recognizing and comparing for identification, like in crime scene investigations and things like that, because that creates that DNA fingerprint that's associated with that. So how do we usually see that displayed when we're looking at CSI is in gel electrophoresis, which is our next topic. So gel electrophoresis is the means by which we're going to separate DNA fragments in a gel, and they are an agarose gel. So we talk about agar, or the sugar that helps with solidifying the agar is called agarose. And so taking just that purified sugar, have a nice clear gel that's just solidified for the sake of basically allowing DNA fragments to move through it. We can do gel electrophoresis with DNA or with proteins, just make the gel out of something else, not agarose if we're doing proteins, but DNA will do it in agarose. And DNA, we've already learned about how DNA Basically has a negative charge. It has a negative charge because of those phosphate groups. Remember, a nucleotide, we would have in DNA, this would be deoxyribose. We would have that attached to a phosphate, which is PO4, and it has a negative charge to it. A negative charge is what's key here. And then that's attached to a nitrogenous base, A, T, C, or G. So we'll just put NB for A, T, C, or G there, right? And it's nice ring structure, purines and pyrimidines and that sort of stuff. So this is an actual nucleotide, which is the building block for our nucleic acids. And that these are negatively charged, which means they're going to go towards the positive pole. Opposites attract. This is what allows those DNA fragments to move towards the positive pole through the gel. But then those fragments will separate based on their sizes. And this is because the larger DNA fragments get stuck and the smaller ones travel faster. Think of it as like a mesh. Small pieces can move faster through the mesh, but the big ones get kind of stuck and it takes them a little bit longer to make their way through. And so that rate of movement basically can create us those gels. And then we can add chemicals or use like UV light and things like that in order to visualize those bands that are on the gel. And we can compare person to person. And so we can see here in this example that we're looking at, if we look at each of these 1, 2, 3, 4, 5, 6, 7, 8 is a different person or a different sample from people and whatever. If you look at person 3 and 4, what do we see here? Almost identical for 3 and 4 on the bands that are generated there. Almost identical. So these might be twins here. So genetically identical, probably identical twins. Okay. So everybody else, though, different people, different fragments might have similar patterns in similar places. Maybe everybody has a band here, but not everybody has all of the exact same bands and at the magnitude that we might see too. So the more specifically that we do this, the more that we can see that. And these all represent different fragments, and that the ones that are down here towards the bottom of the gel are the shorter ones, and the longer ones are towards the top, because the longer ones get stuck, they can't migrate through as fast. The smaller fragments can migrate faster. So we can actually use this to develop a fingerprint of people. DNA is essentially the barcode of life, and those visualization of those bands can create a fingerprint that's unique to you and is repeatable for you each time that we cut you with those same enzymes. And so we can actually use that to compare it from person to person. So we have in the next slide, suspects of a crime and victims of a crime. And we can see evidence from that site. We see evidence, 2 pieces of evidence and the victim's DNA. We can see that we have 2 different suspects and that our evidence samples seem to match suspect number 1. And so we can identify who we think committed a crime based on their DNA fragments from cutting them up with restriction enzymes. We also, if we're going to be studying genetics, we have to remind ourselves when we learned in the previous chapter about introns and exons, that if we want to put like human genes inside a bacteria to have them make our proteins, that bacteria can't splice out the introns and splice together the exons. Remember, exons are the coding regions. Introns are basically just extra bits of information that we don't need to code our proteins. All right. And so in eukaryotic cells, we have to go through the splicing of the exons together and removing the introns. So basically, what we can do is, so this is the normal process for eukaryotic DNA going to precursor mRNA. We need to remove the introns, which is our kind of gray area here, and splice together the yellow area, which is our exons. So then that creates mRNA that our ribosomes would read. Well, what we can do is we can take the cells of a eukaryote like me or you. Let's say we could take the cells of my pancreas that are filled with this processed, spliced mRNA and extract that spliced mRNA, that processed mRNA, and then reverse transcribe it back into DNA, and it no longer has any of those introns in it. And then that creates cDNA, which is complementary DNA. It's complementary to the spliced mRNA product. And then we can put that cDNA into bacteria, which is how we get insulin. We literally took the cDNA of the insulin mRNA, which was already spliced, and took that cDNA and then put that into bacteria. And now bacteria can crank out insulin for us, making it much more affordable for people with diabetes who need insulin. So this is a great example of how genetic engineering can be important and life-saving. Another great tool in the genetic engineering repertoire is PCR. PCR stands for polymerase chain reaction. It's basically a molecular Xerox machine for DNA. It makes copies of DNA. When we say that it amplifies DNA, we mean that it makes copies of the DNA. So you can start with one DNA, double-stranded DNA strand, okay, and basically turn it into two after one cycle of PCR. And then each of these two in another cycle will make another two. And so we consider it to be exponential. And then the next cycle, double each one. So each cycle doubles what you had in the previous cycle, and then basically amplifying, copying the DNA that way. So we need to understand a little bit about the steps of PCR. It is one of the most commonly used tools in like hospitals for diagnostic purposes, and we're going to learn a little bit about how that works. So it uses the same exact events as DNA replication, which is basically opening the double helix, exposing each single strand as a template, and then adding primers. Remember, DNA replication had primers. It was RNA primers. Here in PCR, we can just use DNA primers because we don't need to delete them. We're just seeing if we can elongate the strand from that. And the primers are basically what determines whether you're going to be positive or negative. Yes, if you elongate, you're positive, but you can't elongate unless the primer bound, and the primer won't bind unless the sequence is there. And that is what determines how DNA replication works. And it does use DNA polymerase. And so, again, the primer is probably the most important thing in determining whether you're going to be positive or negative. They are anywhere from 15 to 30 nucleotides long. And basically, if the primer binds, that means that the sequence is there. So if you are positive for a COVID PCR, that means that the primer bound to COVID DNA sequence. Let me draw a little bit of a picture of it. These are your original strands. We will use heat to separate them. So now we have these single strands. Make them equal, okay, whatever. And then the primer matches up. It's 15 to 30. I'm not going to draw it 15 to 30, but they'll match up on each side, okay? If they match up, you have the sequence for COVID. If they don't match up, you won't have any primer to build your nucleotides off of. So what happens next is we would build the next nucleotide and the next one and the next one and the next one and the next one, but we can only do that if the primer was there. If the primer didn't bind, we cannot add nucleotides to nothing. You cannot. Nucleotides have to add on to a primer. So the primer determines whether you're positive or negative, but the machine detects that you're positive or negative because we're adding on nucleotides. Many of the times, whenever you're using PCR as a diagnostic tool, this is done by each nucleotide has some fluorescent molecule that when you add the A, it fluoresces yellow. When you add a T, it fluoresces red. When you add a C, blue. When you add a G, green. Okay? And so each of those nucleotides can flash a color in the system if they're being added on. And so your PCR machine can actually detect each color and tell you how many A's and T's and C's and G's are being added. If you're getting a lot of addition of nucleotides, your primer must have bound, and therefore you are positive. It's just a matter of whether you're elongating or not. And you're only elongating if the primer bound. If the primer did not bind, you will not elongate, and then you're negative. So it's just looking for that sequence that you have the primer for. That primer only binds to COVID genes, or it only binds to Staphylococcus aureus genes, or it only binds to genes associated with the flu. They're very, very specific because they're 15 to 30 nucleotide bases that are unique to those genes for that organism, for example. Because we're going to be separating our DNA using heat, we have to have a DNA polymerase that isn't going to be denatured from heat. Remember, any hydrogen bond can be denatured with heat. And so that would also denature the proteins, including DNA polymerase, which is a protein. Enzymes are proteins. And so we have to have a heat-stable DNA polymerase, which we got from thermophilic bacteria that live in like boiling water vents. And so we stole it from them. Okay, so cool stuff. So there are only 3 steps to PCR. PCR's 3 steps are denaturation, priming, and extension. It's just like I drew out, but denaturation is going from that double strand, adding heat to the 2 single strands. This is denaturation. Then taking those 2 single strands and then binding the primers like we did before is priming. Okay. And then extension, like we had before, would be adding on the nucleotides from that primer edge in the 5' to 3' direction. It always works in the 5' to 3' direction, adding on to an existing primer. So that third part. And that's it. And then heat would separate those new ones and then make new. And then this is an infographic basically explaining that. Having your first double-stranded DNA, separating. That was the first step. So this was after that first step. Then adding our primer is the second step. And then adding the nucleotides in that third step. And then separate again. And then prime again. And then elongate again. And just repeat that process. Every step of PCR is doubling what you had in the previous one. So in one cycle of PCR, if you had 1 piece of DNA, you became 2 pieces. In the second cycle, if you have 2, then you became 4. In a third cycle, if you had 4, you became 8. In a fourth cycle, if you had 8, you become 16. In a fifth cycle, if you had 16, you become 32. And so on and so forth. Doesn't take very many cycles to get up to a high number. So the idea here essentially is that you can have like 30 to 40 cycles and you'll have like millions or billions of DNA starting from just one double-stranded DNA. And if you are continuing to do this, elongate cycle after cycle after cycle after cycle, then you are positive for whatever that primer binds to. We can also use this to take a little bit of a target DNA and make more copies of it for use in genetic engineering. Again, if I mix a little bit of cut out, genes from pancreas, and I want it to stick into a plasmid to put genes into a bacteria, then I'm going to need a lot of that gene so that it favors binding to the gene instead of binding back to itself with those restriction endonuclease enzyme cuts, those sticky ends that we talked about earlier. All right, so there's a video about this that I can't play it in the recording, but I highly recommend that you guys go back and watch this that basically walks you through how PCR works. Alrighty, so this whole concept of how we're going to be using copies of DNA, restriction enzymes, sticking DNA to each other and making new DNA as a result of it, this is recombinant DNA technology. So anytime that we are doing this, we usually will have, we call this whole process cloning, where we'll remove a gene from an animal or an organism and then put it into a vector. Often this will be a plasmid, which again is just a circular bit of DNA that has extra genes in it, or it could be a virus, and then using that virus to stick new genes into places. We'll then insert the plasmid or the virus, or whatever, into a cloning host. We often will use bacteria like E. coli, it's commonly used, or yeast, which would be a eukaryote, which might be able to process genes if we didn't want to do that. So then that gene is then translated by that cloning host as a result of that. Oftentimes, we will remove DNA from targets, cut them into pieces with endonucleases, and then stick them back together and seal them again using DNA ligase. If we need more of DNA in order to work with it properly, then we can amplify that DNA using PCR. Again, plasmids, circular bits of DNA that can be basically altered by cutting with restriction enzymes. And they often are engineered to have multiple restriction enzyme sites. So depending on what gene or what sequences you're dealing with, you might want to use different enzyme sites to cut the plasmid, depending on what you're working with. You can also use bacteriophages in order to insert genes through that lysogeny process that we learned about in chapter 7. Anyways, so this is an example of a plasmid. This is PUC19. The P stands for plasmid. And basically, this is just a vector that we could insert genes anywhere at those restriction sites. What's a restriction site? Again, this is it. This is a site that can be cut by the enzyme EcoR1, which we talked about previously. And each of these is a different site that can be cut. They have a different palindromic sequence there, cut with different enzymes, depending on what you're trying to do or what sequence works best for whatever your application is. And so this can actually help you cut very specifically and even orient your gene in certain ways, depending on whatever you're trying to do with this. So this is all done very thoughtfully by genetic engineers whenever we are inserting genes in there and into our plasmid. It's also important, we'll learn about this in lab 10 when we talk about transformation, but basically it might have ampicillin resistance. Your cloning vector, your actual your plasmid has antibiotic resistance. So that when we put the plasmid into the host, the cloning host, E. coli, for example, normally E. coli would be killed by ampicillin, an antibiotic, but now we've made it resistant. So that's how we know if it grows on media that contains ampicillin, it must have this plasmid. And so that tells you that that organism took up that DNA from its external environment, which is called transformation. It's a horizontal gene transfer method, which we learned about in the previous chapter, chapter 6. All right. All right. So again, if you're sticking fragments into other DNA, you'll have to seal it back together. Seal those sticky ends using DNA ligase to reform those phosphodiester bonds, which are the bonds between the nucleotides. All right. This also can rely on the use of reverse transcriptase. Reverse transcriptase is an enzyme that basically turns RNA back into DNA. So I told you, if I take processed RNA from my pancreas cells that's already been spliced, we already brought those exons together and removed those introns. In order to turn RNA back into DNA, that's a process that our body can't do. That's something that we didn't think existed until we discovered HIV and other retroviruses produce this special enzyme called reverse transcriptase that basically we normally go from DNA to RNA and then make protein by reading the RNA, right? We learned about that in the previous chapter, that in order to go from RNA back to DNA, this is the reverse process. So it is reverse transcription. The enzyme that does reverse transcription is called reverse transcriptase. ACE often signals that it's an enzyme. So this allows us to make DNA from RNA. So remember, if we do that, that would be complementary DNA or cDNA, which we can then put into a cloning vector like a plasmid. So then we can put that gene inside that plasmid and put it into the bacteria. Again, we wanted to do that with sticky ends using restriction enzymes, and this is what it would look like. So basically, what would happen is you would have the donor gene sequence. Here are our restriction enzyme sites where we're going to cut at the palindromic sequences, making those sticky ends. Make a similar sequence in a plasmid, making more sticky ends. And then we mix them together in excess. So lots and lots of the gene and some of the plasmid. And so that the plasmids will, some of them, allow us to stick this gene into the plasmid. And now we've made a recombinant plasmid that contains the gene. So this draws out that whole picture. Then we can take that and put it into a bacterial cell. And that cell will make mRNA, which will then translate it into a protein product, and we can filter it out from like the broth or whatever we're growing that organism in. So that is the idea of how PCR and restriction enzymes and everything like that can be used to make, genetic recombinants for use in medicine and investigating things in science and everything like that. Our next concept that we'll talk about in this chapter is nucleic acid hybridization. Again, if you have a DNA sequence that is sitting there, you can imagine that if the primer only binds to that specific sequence, do we always need to amplify the DNA, or can we just see if the primer bound? And that's the idea with nucleic acid hybridization. Again, you make a probe that matches whatever your gene sequence is. If the probe binds to the sequence is there, and we can even label that probe with fluorescence, for example, in order for us to be able to identify. So this looks for genes based on DNA matching up, being complementary. If the complementary sequence is there, then the DNA sequence is there and it is positive. So this doesn't require electrophoresis. It doesn't require amplifying that sequence either. This just sees if the gene is there. And this is nucleic acid hybridization. So the idea of how this actually works in practice is that you would have a plate with colonies that we don't know what it is. We would grow it up, and then we would get a little filter paper that we smashed onto our little plates, just like we have in our lab. And then it gets some of those colonies on it. We then treat it to break open the cells and break apart the DNA. And then we bind our probes that have fluorescence on them. If the probe binds, then those colonies have the sequence that you're looking for. Those colonies are the ones that are staph or are E. coli or whatever. So we can use this for diagnostic as well as for identification purposes in general. If we're using fluorescence and we're using live colonies or cells that are basically original cells, then this is called fluorescent in situ hybridization. We'll learn more about this. This will come up in a diagnostic aspect in future chapters. We'll talk about this in our diagnostic chapters. But basically, this is the idea of using that nucleic acid hybridization, matching up DNA sequences of single-stranded DNA with a probe, and then linking that probe, that DNA sequence, to a fluorescent marker, and then just seeing if it's present in the cells or not. So this is fluorescent in situ. In situ means in like the situation, in the actual cells that are still intact typically. And then looking for the presence of those genes. So we use this to detect DNA or RNA in cells or in tissues even, which is useful for looking for certain mutations associated with certain diseases. In fact, we can make chips that have a whole array of examples of different genes and then take the patient sample and see which of the spots on the chip that it hybridized with. And so DNA microarrays, like using nucleic acid hybridization, but for a lot of different kinds of genes. It's very expensive, but if you need like a shotgun approach to looking for different mutations or certain bacteria or something like that, then these microarrays, which is just a tray full of a whole lot of examples of genes, would allow you to look for that sort of a thing. Very sensitive and can be used for identifying different subtypes of cancer. You know that this patient has breast cancer, but what type of mutation led to that breast cancer? Microarrays can be really useful for identifying that sort of stuff. Also looking at genetics and how we use it, we can use DNA profiling, so like DNA fingerprinting, sort of like using those restriction enzymes to create your DNA fingerprint. We can create fingerprints for different organisms, and that can help with identifying or for identifying individuals as well. So like suspects in cases and things like that, which we talked about previously. We can take this even further and do genome analysis, taking those restriction maps or literally figuring out what the nucleic acid sequence is for an entire genome of an organism. And then we can use that for further studying and science and things like that. So that's the idea of how sequence maps would be made, not just based on restriction enzyme fingerprints, but taking it for the actual nucleic acid sequence, the GATC of it all. That's how sequence maps would be made. These can be useful for studying organisms, especially as you're learning more about their genes and what their genes code for, especially as predictive aspects in basically, biology in general. Okay, so we can essentially make a map of all the genes in an organism and predict what those genes are going to be coding for. And that can give us more information about that organism and what we can predict about that organism overall. All right, moving on to single nucleotide polymorphisms. A single nucleotide polymorphism basically is saying that a single nucleotide can cause a change in a protein that can lead to a wildly different protein outcome. Essentially, this goes back to chapter 6, where we said that AAA and AAU might code for the same amino acid, but then AAC might be a different. And then, I don't know, UAA might be a stop or something. So this is talking about that, like, one nucleotide, this is your original, one nucleotide might be no change, or it might be a different amino acid, or if it's the first nucleotide, then it can lead to a stop, or whatever, depending on what organism we're talking about. So one nucleotide can be changed to lead to a different outcome. Those are single nucleotide polymorphisms. Okay, the human genome contains 10 million single nucleotide polymorphisms. That means that in organisms, we have seen one nucleotide leading to a different outcome 10 million times in all of human genetics while doing the Human Genome Project. So this is the idea. One nucleotide change leads to a different outcome. That could be as simple as a different color of hair, but it could also be as dangerous as death or lack of survivability, inability to create a living organism. It would never make it to, the fetal stage. So thrombophilia is this idea for a clotting factor where we would have excessive clotting, that we had one nucleotide change where it's normally going to be GCT, normal, what we call normal functional version of that gene. Instead of GCT, it got changed to GTT. One nucleotide got changed. That changed the amino acid. Instead of arginine, we now have glutamine. Because we have glutamine, that means that protein leads to excessive clotting. The protein is formed incorrectly, and then it has too much clotting as opposed to the appropriate amount of clotting. So that's an example of a single nucleotide polymorphism that could be dangerous for an organism. All right, there's a lot of different things we can get from all of this cloning and scientific analysis and things like that. Basically, looking at our recombinant DNA technology, allowing us to make recombinant organisms that have different genes in them, different protein products, and looking at the effects of those proteins and how that affects the organism. And all this can be used to study science and how biology works, or it can be used to make things that help us either in medicine or food or things like that. For example, making insulin or human growth hormone are all products of recombinant DNA technology. We have a lot of different medications that are used this way. We can use interferons, interleukins, or tumor necrosis factors that are made through recombinant technology to treat things, including treating cancer or regulating the immune response and things like that. We have ability to make recombinant DNA technology for creating things like Remicade or Humira, which are biologics, which are basically made to dampen the immune response against yourself. So treating autoimmune diseases and other things like that we have generated through these technologies, including vaccines and other things. Anytime that we have an organism whose DNA we've manipulated in this way. So when we have bacteria making human insulin, that's a genetically modified organism. The other word for genetically modified organism is transgenic organism. They are the same thing. GMO and transgenic are the same. So GMOs, genetically modified organisms, are transgenic. Means that their genes, their genetic makeup has been altered some way by science. Okay. So we have microbes, plants, and even animals that have been altered to become genetically modified. If we have directly tampered with their DNA, they're genetically modified. Okay. This includes certain bacteria that have genes in them that can basically be made to not freeze from freezing and stuff like that. We have certain genetics in certain bacteria that can basically fight off insects, that we can put those genes into plants and then make them resistant to insects. And so most corn that we eat nowadays, almost all corn that we eat nowadays is genetically modified to have this bacteria, Bacillus thuringiensis, has a gene that basically makes it resistant to insects. And then that's how, we've modified those plants. There's a problem with this. First of all, you eat the corn, doesn't change your DNA. You eat DNA all the time. If you eat a plant or an animal product, You eat DNA. I don't care if it's GMO or not. It had DNA in its cells. You eat it, your body breaks it down. There's no risk of these genes causing your genes to change. That's not the problem with GMOs. The problem with GMOs is that plants can exchange genetic information with one another through a process called transposons, basically jumping DNA. And if you have bug resistance in corn, for example, that could then jump to weeds or natural plants. Now bugs don't have anything that they can eat, and that ruins the ecosystem, especially organisms that depended on the bugs for the food chain and stuff like that. So the DNA can jump, but not into you, into other plants. And then that is an ecosystem liability that we're talking about. So that we don't see a ton of that, but it has happened. There is a trend that suggests that this could be a bigger issue going on. But GMO organisms do not transfer the genes to animals that consume the products. That's not the issue. The issue is the plant transfer out in the wild, and then how that affects the ecosystem. This also leads into the concept of gene therapy. So this is the idea of, this is the idea that we can basically alter the genetic makeup of organisms intentionally, including you. And so we could use some of this technology in order to change your genetic makeup. I'm not talking about changing your eye color. This is a realistic scenario for cosmetic sorts of issues like that. This is more along the lines of illnesses that are related to your genetics that are potentially deadly over time. So if you carry a certain gene that predisposes you to a certain issue, bring back the slide, to a certain issue, we could potentially change that gene in your genetic makeup. There are a couple of different versions of gene therapy that we need to be aware of. There is gene therapy that affects your regular body cells, and then gene therapy that affects your gametes, your eggs and your sperm, that can then be transmitted from the future. So we basically can also use this. We can put the gene into something like HIV. HIV then inserts that gene into your body, and then now your body produces that gene naturally. And we can even target the cells that it inserts the gene into, especially if we're targeting the bone marrow. We can remove your bone marrow, infect it with this HIV virus that has this new working version of the gene. And then now you have the working version of the gene because the virus gave it to you. Now, you might be sitting here thinking, I'm not going to let you infect me with HIV. It's not HIV anymore, boo-boo. It's not HIV. We are smart enough. We know we should remove the HIV aspects of it. We can still use the virus, hollow it out, and just put this gene in there. So it only inserts this gene, not HIV genes. And so we have altered this. And this is used in about 13 different genetic diseases now and does seem to be effective. It's not 100% effective for everybody, though. There are some people that the virus doesn't take, it doesn't infect, or for some reason the genetic doesn't stay in the cell for some reason. And so there's further investigation into that. But there have been several instances where we've been able to use a virus to insert working version of that DNA into your DNA. And now you can make a version of that gene that you couldn't before and actually treat diseases. So what kinds of diseases? Spinal muscular atrophy, inherited vision loss, acute lymphoblastic leukemia. And again, if it's only affecting your body cells, your bone marrow is a body cell. Is it a stem cell? Yes. But does your bone marrow, is it involved in making babies and spreading on the gene onto your offspring? No. So if it can't be spread onto the offspring, that's somatic. If it's in the body cells, it changes just those cells in just that organism. It is not going to get passed on to offspring. So the offspring would still get the bad versions of the gene. If we're actually doing gene therapy that can be passed on, then we must do it at the embryo stage or the egg or the sperm stage. Then that would be called germline therapy, and that is heritable. But again, that would have to be done at the embryo stage. We have to change the genes at the embryo stage, not as in an adult. Right now, gene therapy only works in the somatic cells for an adult that have, adults that have, or even children. If you've been born, this is what I'm talking about, okay? Somatic changes, body cell, not heritable. All right, so that sums up chapter 8. And if you guys have any questions, let me know. Otherwise, I will see you for the next one. Thanks.